goes on, we've got the invading fetal trophoblast here. It's not expressing HLA-A or B. We think that's probably a good idea because the mother's immune system might react to the paternal A and B on the trophoblast, on the fetus. But we know it does express HLA-E and HLA-C. These are all good ligands for NK cells, and HLA-G. And the thought is that interaction of the of activating KIA receptors on the uterine NK cells of the mother with the ligands on the invading trophoblast causes activation of the NK cells, which then secretes interferon gamma, which is absolutely critical to initiation of the modification of these spiral arteries. So if there's inadequate interaction between the uterine NK cells and the ligands on the trophoblast, inadequate modification of the maternal blood vessels. So this is data from Ashley Moffat's group looking at the proportion of controls or patients with preeclampsia who are homozygous for the KIA A haplotype. So these mothers don't have any activating genes, if you like. It's only a small difference, so sort of 25% in controls and about 35% in the patient. She had large numbers, so it's a significant difference. But if she then subdivides those pregnancies into those where the fetus has HLA-C2 on the left here, and those pregnancies where the fetus does not have C2, the C2 ligand, she now finds that the frequency of homozygosity for the A haplotype is now considerably increased in those mothers who experience preeclampsia compared to controls. And I think, if I'm probably not misquoting Ashley Moffat now to say that, well, this was done in terms of homozygosity for the A haplotype. I think she thinks now that it's actually the KIA 2DS1 activating gene on the B haplotype, which is important for interaction with HLA-C2 on the fetus. It may not, that may be an oversimplification, but I think that's where she...
That is missing. That would be missing in the mother. The 2DS1 would be missing in the mother.
Yes.
If it's A homozygous.
Yeah. So you need the 2DS1 if the fetus has C2. And she showed a rather interesting graph. Epidemiologically, we know that the frequency of preeclampsia is pretty much the same all around the world. But we know that the frequency of what's this average? Frequency of C2 varies greatly from one population to another. So why isn't there a correspondingly different frequency of preeclampsia in these populations? So what she's seeing here, showing here, is that in those populations where C2 is very high, the frequency of the A haplotype, so this is the opposite of 2DS1. So the frequency of the A haplotype is very low, or the frequency of the B haplotype, if you like, is very high. 2DS1 is high. So there seems to be this inverse relationship between the frequency of C2 and the frequency of the A haplotype in the population. That might be why the frequency of preeclampsia is maintained at the same frequency despite different frequencies in C2. Okay, I think we are running out of time. It would be a shame not to mention that there have been reports of KIA association with other diseases. So this is looking at the effect of the presence of 3DS1 and BW4, the two together having a synergistic effect on the outcome of HIV infection. So this is the proportion of patients who are AIDS-free at this time after HIV infection. So those patients who have 3DS1 and BW4 seem to have a sort of protective effect. And the second viral disease, hepatitis C, being homozygous for 2DL3 and homozygous for HLA-C alleles with the C1 epitope seems to enable you to clear HIV, Hep C virus from the liver. And I think we have run out of time, so I'll wind up there. Okay.
Thank you very much, Campbell. Are there any burning questions? Of course, several of the students will talk after the coffee break on the same topic, so maybe the things which are not clear yet will be answered by them. But if you have burning questions to Campbell, we have, I think, five minutes for questions. Yes?
Thank you very much for a very informative lecture. And just I have one question about autoimmune diseases. Don't you think the situation of autoimmune diseases and care might be worth investigating more than the transplantation?
Might be worth
investigating, especially with haplotypes. Do you think there will be a role for A or B haplotypes with certain autoimmune diseases like diabetes?
People have looked. In diabetes, in fact, people have looked. In fact, I think there is a publication from Leiden, is there not, France, on frequency of particular care genes in type 1 diabetes. In general, they seem to be, when they have been reported, a positive association, the effect seems to be fairly small, and I don't think a lot of those diseases in which associations have been reported have been replicated. So I think it seems like a reasonable thing to investigate, but so far the data hasn't been all that impressive.
Can the system be triggered by soluble peptides?
You can repeat?
Could you trigger the system by soluble HLA peptides? Yes. Could you use synthetic peptides to block the NKE effect?
So the question is whether these receptors are also susceptible for soluble HLA molecules, and whether you can interfere.
In soluble form. Yes. Could you use synthetic peptides to block them?
Theoretically, I would say the answer is yes. I'm just trying to remember whether I can remember any publications in which that has been done. I don't think I can, but it seems like a perfectly reasonable proposition to me. Can you guys remember anything on soluble?
I think people tried, but it didn't work.
Really?
Yeah. Was it HLA-G at certain point docked?
Yeah,
the soluble HLA-G.
The one exception to that is soluble HLA-G, which interacts with KIR2DL4, which is my favourite receptor. So I believe we have now shown, Marcello, that soluble HLA-G does not interact with 2DL4, but what it does do, it does interact if your purified NK cells are slightly contaminated with dendritic cells. And we think the soluble HLA-G is binding to ILT4 on the DCs, which is then presenting the 2DL4 to HLA-G to 2DL4.
But I remember that somebody did experiments with, say, clones recognizing C1 or C2 and had serum of people with the proper HLA type and see whether you saw a difference in the reactivity of these clones. And the soluble C1 or C2 could not block response.
But you probably need to cross-link the receptor, so you have to immobilize the ligand.
Yeah, you may need solid phase to cross-link the key receptors.
Quick question. In the transplant key ligand mismatch, in the haplotype mismatches, people look at group 1, group 2, and BW4. What do you think about A3? Because Vanderson uses A3 as well in the Corbats.
People have not usually analysed KIR3DL2 and A3A11. I don't know why they haven't. I guess there's been a lot of uncertainty about whether A3 and A11 are really the ligand. I think it's pretty clear they are, but there are a couple of papers indicating that arming of NK cells that express 3DL2 is not very strong. They seem to be rather